Good afternoon, and thank you very much for being here. It's a great pleasure for me to tell you about what we're doing in this context that Dimitri already introduced. So the experiments that we are doing, aiming at building such Maxwell demon, are based on what we call single molecule experiments. So what I will first do is give a general overview of what these experiments are, and then show you the application for these such of non-equilibrium physics studies. I will start reminding that single molecule methods are remarkable because they are techniques developed over the past, let's say, 20, 30 years that allow us to manipulate with high precision small objects such as molecules. And molecules are particularly interesting because they contain lots of information. Biological molecules have sequences, and therefore they code for information, and therefore we can understand, for example, how this information is used for specific biological functions. By measuring molecules one at a time, we can track with high resolution all the energetics of the processes where such molecules are involved. So that would be I would say, the most relevant application until now that I will focus in this talk, which is that we can explore the energetics of small systems where fluctuations are so important. There is another aspect that I like a lot, which is the fact that single molecule methods are ideal to explore diversity in samples, which is, so in biology it's very important, the fact that energy, we know, is a driving force of basically everything, but in biology, energy is nothing by itself. It needs to be channeled, fueled in the right direction, and therefore it's important to be aware that in biology, diversity is a way in which such energy is somehow encoded. So information in biology is very relevant, and we have few doubts that many processes in which in biological beings, energy, any source of energy is somehow fueled into information, okay? And therefore, these single molecule techniques allow us to somehow characterize such diversity, okay? This heterogeneity of these ensembles, these biological ensembles. Not two cells are ever identical, even if they are grown from the same strain. And that's something that must be taken into account. The techniques that we use for single molecule, there are several techniques for single molecule manipulation. The one that we use in my lab is optical tweezers, which use light, okay, to focus in a spot in water. In water, that's a fluidic chamber usually, where you have water inside, you have your molecules, and by focusing that light in water, you generate a spot. You can capture a micron-sized bit, and that's a force probe. You can use that force probe to manipulate, to exert forces into molecules, but you can also use that probe to measure the forces themselves. So you will use the force probe to manipulate and to exert forces and to measure them. So that's that's the illustration, the picture, the schematics of the instrument that we have in my lab, which are counterpropagating beams to generate a stronger gradient capturing forces. And the setup is showing here the experimental illustration of the setup, how we pull a molecule. So you have one bit in a pipette, one bit in the optical trap, and the tether in between. Okay? So which type of experiments you can do with this optical tweezers instrument or this single molecule technique? You can measure elasticity of polymers. That has been very important because it's another way to probe the physical properties of polymers. We can understand, we can contribute somehow to biochemistry by characterizing the energetics of molecular interactions, such as folding energies, binding energies, affinities, and so on. We can also track enzymatic activity in biochemical reactions, okay, by measuring the translocation of molecular motors. And I would like to put this last aspect in here, non-equilibrium physics in general, and in particular, one of the aspects of this non-equilibrium physics, which is this information to energy conversion or to work conversion that has been discussed by Dimitri on the basis of macrodemon, and which is at the core of the project that we are discussing today. Okay? So let me just, as an illustration for those of you who have never seen such experiment, this is a typical experiment that nowadays is a classical, but is used even for calibrating force measurements. This is called DNA overstretching. So you have two bits, as I said, one bit in the optical trap where we exert the force and measure the force, and one bit which is bound to a pipette, so it's a surface, it's a fixed surface. So the idea is to establish a tether molecule between the two bits, move the optical trap relative to the pipette, and exert force on the molecule, tether between the two bits, and measure it. Okay? So here I will show you the most basic experiment, which is, as I said, DNA overstretching. It's already 20 years back, this classical experiment. These were two papers were published back to back in Science, where they will show how they can measure pulling forces and responses of a DNA molecule, double helix, up to 100 piconewtons approximately. So what you do is you synthesize, you express your molecules, you prepare your sample of DNA molecules, you label them with small chemical groups, such as biotin and sort of antigen molecules that can bind specifically to antibodies. You code one of the bits with antibodies to bind to this antigen here. You code this bit with equivalent of the antibody for the biotin, which is this streptavidin, and then you code your bits, you label your DNA, you mix everything in the chamber, and then you establish a tether. So the procedure to the experiment, you first have to synthesize your DNA molecule. That's not very important how we do, but we use typical methods of very simple methods from biochemistry to take a DNA molecule like lambda DNA, which is from a phage virus, which is 48,000 base pairs. We cut into halves to have a shorter molecule because we can have a range in our instrument of about 10 microns. So 48,000 base pairs would be approximately 16 microns of control length, which is too large for a range of our moving pieces. We cut it in half, so we have a shorter molecule available for pulling. We label it, and then that's the experiment. What I will show you here is a movie of how it works. So here you see the pipette, and here you see this light, this illuminated point. There is a filter here. There is optical trap. It's too strong, the light, to show you here in the animation. So we have to put a filter so you can see the spot without being too shiny. Because now we're going to capture bits. The first thing we are going to do is we're going to trap a bit that is supplied by a channel to the central channel of the fluidic chamber. So one type of bits will be supplied by one tube, one channel, and will be trapped in the optical trap, and then will be brought to the tip of the pipette. And the other bit, which is coated with DNA and with antibody and so on, the other bit will capture, being supplied from another tube, and being captured in the trap. And then we will approach the two bits together to the experiment. So, so here, let me see that I fix. It's always difficult to make this start. Okay, here it is. So you will see how we move the chamber. We move the fluidic chamber to position the optical trap at the end of one of the supplier tube. There are bits coming out. You see one bit here, and at some point you will see how one bit, which is 2 micron of diameter, is capturing the trap. Now we move the fluidic chamber. We position on the tip of the pipette, and we mobilize by air suction. There is a syringe that does sucks air, and then immobilizes the bit. Now this is fixed, and then we look for the other bit, which has been incubated with the DNA. So the DNA is labeled at the end, and apparently it's bound to the surface, but it's free on the other end. So there are hairs protruding from the surface of the bit. Of course, you don't see them because DNA, even if it's 8 microns control length, it's very thin. It's 2 nanometers of diameter. And now you have here the DNA protruding out of the bit, and then we have to fish the end of the DNA. Now, the end of the DNA is labeled with biotin that should bind to the streptavidin that is coated this bit in the tip of the pipette. So it's fishing, the procedure is to fish. So that's how we establish a tether. We play with a mouse, and then we wait for some time until we get something. So now you will see typically, because DNA is a random coil, it's 8 microns control length, but it's compact in a random coil. You have to approach here you see we have the tether. So here is a typical thing, and now we have a photodetector positioned at the other side of the illuminating laser, and with this photodetector we can measure the deflection of light, and we have a way to extract directly the force acting on the bead, which is the force acting on the tether. Okay? So this is the single molecule setup. When you do this, then you can get these measurements, which is a so-called force extension curve. That's what single molecule biophysicists aim for. Try to get this data. This is actually very good data. So it goes from 0 to 80 piconewtons, and here you see how the force goes up. That's what we call the entropic elastic regime. And this response, forces versus molecular extension, which is the distance between the two beads, gives us a measurement of the persistent length of the double-stranded DNA, which is a quantity important to characterize the bending rigidity of a polymer, which is otherwise very difficult to measure with other methods. You can also measure the Young modulus, which gives you the free energy, the contribution to free energy of stretching the DNA. But what was important is 95, when this experiment was done in 95 by the group of François Caron and Carlos Bustamante, okay, they discovered this remarkable transition at 767 approximately piconewtons, which is this plateauing force, which is known to correspond to a conformational transition of DNA. Of course, this occurs at so high forces that no one had observed previously in biological, in a biological setup, because those forces are pretty high. I mean, you have to take into account that 70 piconewtons is high. I mean, molecular motors, such as, for example, those motors that intervene for packaging DNA inside infected cells by virus, exert up to 60 piconewtons. These are the highest because they have a huge pressure they have to overcome to inject the DNA inside the capsids. So it's really remarkable force. Now, this was one experiment which is stretching DNA, but I will show you, and that goes to my main talk here, which is the fact that we can use these measurements to explore fluctuations in the small systems. We can do another type of experiment. This experiment, we pull DNA from the two ends. Now, what I will do is slightly different. I will pull DNA from the same end of the two strands. So the experiment is totally different. Okay? It's like if you stretch a double cord, you stretch it, or you pull from the same end a double cord, then you will unzip the double cord and get two strands. And because DNA is a double helix, that's what will happen. So now the experiment is different, and it's called unzipping. And here you will see what happens. This is a response force versus extension for pulling from the two different ends. If I start stretching DNA from the same end, the two strands, I get unzipping. I unzip the double helix. I denaturalize. I melt, if you want, DNA using mechanical force rather than temperature. Okay? And that's the response that you get. Okay? That's stretching this molecule, which is about 6,007 microns approximately until here. And you get the sawtooth pattern corresponding to the breaking of all the complementary base pairs. At the end, you get the elastic response of the remaining single-stranded DNA. And when you get back, you release the force, and remarkably enough, the molecule nucleates at the center and forms the double helix again without any apparent irreversible behavior. So there are a few examples of fracture processes so reversible as this is. Okay? So this is a remarkable reversible process. The blue curve superimposed to the red curve. And why this is so? This is so because nature or molecular interactions are weak interactions. They are about kcal per mole. If you take any book of biochemistry, look for energies of binding, of affinities, or anything. Proteins binding DNA, DNA hybridizing with itself, enzymes, I don't know, binding to other enzymes, forming big complexes, you will see that the units are kcal per mole. Now, 1 kcal per mole at room temperature, which is where biology takes place, is about approximately 1 kBt. Okay, it's a bit more. Okay, it's 1.6 kBt. Okay, so this means that the energies that are involved in breaking the bonds of the double-stranded DNA are around kBts. So it's clear that you are fracturing something where the thermal energy of the bath gives you enough heat by fluctuations to allow for these systems to break and recompone, so to unzip and rezip in a quasi-reversible way. Okay? We are at the borderline, or we are at the edge of this thermal fluctuations level. You can then, so you may ask whether, okay, this is nice and so on, but this is useful. It is useful, okay? These force techniques by single molecule manipulation give you very, very accurate information. So for example, I show you here the same experiment as before, okay? That's the unzipping of the 7 micron, which is about 7,000 base pairs molecule. And here you see 3 colors. The experiment is shown in black. It's the same curve as before, but before there were fluctuations. So this was filtered out to 10 Hertz, okay, because our data before was 1 kilohertz frequency, acquisition frequency. And if you, for example, well, so if you filter to 10 Hertz, you see the black curve. Now, what we did is there is a model for which you can predict this unzipping pattern. Why? Because the hybridization free energies of the free energy of formation of complementary base pairs have been measured by biochemists since decades. And they know these numbers. In fact, they can predict very well what they call melting temperature. So at which temperature a sequence of DNA will melt. And from this data, because we're doing a melting with force but following a different pathway, we can, and thermodynamics, of course, thermodynamics can be described in terms of state functions. I don't care if I do with temperature or with force. I can reconnect different pathways by knowing the state functions. If I know the free energy of hybridization of base pairs from temperature measurements, I can extract what I would see when I do force unzipping at room temperature, which is what I'm doing. So we can predict using these numbers that biochemists have estimated or have extracted from melting temperature data. I can estimate what would come out. And that's what we found here, which is a bell curve. And what we did, and that was one of the projects we did, is we somehow extracted which were the optimal free energy numbers of hybridization, of binding of complementary base pairs that fits our experimental data. So we found this discrepancy with currently known data, okay? And we could match very well our experimental data. And we discovered that the differences we need to readjust these free energy numbers were by tenths of kcal per mole, 0.2, 0.3, which is very small. And with melting, with bulk methods, with calorimetry, you cannot have such resolution of tenths of kcal per mole. So, but force techniques can, okay? So this is our best estimation, and we could provide numbers for these hybridization free energies. This is just to mention that we can do the same experiment in other conditions. This was sodium chloride, this is magnesium chloride, that's not important. But these numbers that we published back in 2010 were incorporated in servers. So servers, there are servers that predict, let's say, the melting curves of DNA products. So you, for example, give a DNA sequence and you ask them, okay, what is the circular dichroism signal as a function of temperature for my sequence? And they use these sort of numbers that I told you before, these hybridization numbers, and predict you the curve. And now if you go to look at this server that is somewhere in US, you get that you can choose different set of numbers, and this is ours here, Huguet et al. From 2010. That's our numbers here. So these have been incorporated by biochemists, okay? Now, because this is introductory, I must tell you what we can do with these experiments beyond extracting free energy numbers. We can investigate, for example, different sort of molecular structure, easy ones, more complicated ones. We can go from DNA to proteins through RNA, okay? So we can do the experiment with DNA, I already showed you this. We can also mechanically unfold RNAs, and we can also mechanically unfold proteins. So we have a tool that allows us to investigate a wide complexity of biological structures. We could even study cells, biological cells, to study, for example, the mechanical properties of cell membranes, okay? These experiments are complicated, but certainly can be done. So for example, if you pull an RNA hairpin, that's an RNA hairpin, okay? It's a double short, because RNA exists a difference with DNA, which is a double-stranded molecule. RNA exists in single-stranded form, not in double-stranded form. But locally, RNA is like a big flower, it's a tree. It can form locally like structures where it forms some short helices. So RNA has a secondary structure which is remarkably, I would say, complicated. It's not easy, okay? But part of these secondary structures can form some helices. When we have a short helix, we call a hairpin. Okay? So here, for example, you see, in contrast to the previous DNA that was a long DNA molecule, 7,000 base pairs, here we have a hairpin of 20 base pairs. So we can pull on it, putting some molecular handles that are done of RNA-DNA hybrid duplexes here and there. That's just a problem how to express this RNA molecule to pull with our instrument. And then here you see the force versus trap position or molecular extension curve. You see this unzipping, this short signal. This is 2 piconewtons, so this drop is about approximately half this amount. So it's 1 piconewton, which corresponds to the release of the 20 base pairs. Okay, so this is one sort of experiment we can do. We can also pull on proteins. So you can do the same. That's much more, that's more complicated because you have to do the chemistry of linking a polypeptide change, the N and C termini to double-stranded, in this case, DNA handles using disulfide bonds. But you can make this construct and you can pull, and you see the signal. Okay, you see you pull. That's a red curve. There is an unfolding, and at some point you get a plateau where the handles overstretch. So the handles form part of the molecular construct, and it's useful to have them because when you see the overstretching around 60-something piconewtons of the handles, you know that you have one protein in the middle. So that's a good signature of these experiments. You always get many tethers, you don't know what they are. I mean, it's difficult to get proper experiments to isolate what is the true signal of the protein and what is dirtiness. Okay, so that is also part of the art of the experimentalist. To recognize a pattern for something. When you recognize a pattern of something, then you can start having good data. And you can also measure the motion of molecular motors activity. That's an helicase, for example, GP41. This helicase, well, is part, let's say, of the machinery of infection of bacteriophage T4, which is a virus of bacteria. And what it does is an hexametic helicase. So 6 monomers of this complex self-assemble around single-stranded DNA and start to unzip the DNA. So it's the same I did before mechanically, but it is powered by ATP hydrolysis. So it's the same experiment I showed you before that I unzip mechanically, but this is done by the helicase. So what you can do is you can take, in this case, is a hairpin of 1,200 base pairs, so it's shorter, around 1.2 microns. You can unzip first mechanically, and this is the black curve, so this is the standard experiment. This is reversible, as I told you before. And now what you do is at some point you leave the hairpin partially open, for example, at this position. So you leave the last 100 base pairs that unfolded, unzipped, and then you flow a blocking oligo that retains this position. Otherwise, the hairpin would close again back at forces around, for example, 12 piconewtons. So if you don't want the hairpin to close back again and you want the helicase to do the function, okay, you put this blocking oligo that prevents a reformation of the double helix from here to the rest, and then the helicase is flown to the chamber, it self-assembles into single-stranded DNA, and it starts activity. So you can see here the measurement of such enzymatic activity, the position of the trap by a force feedback, by a force clamp of 12 piconewtons. So we move the optical trap to keep always the force equal to around 12 piconewtons. And then what it does is when the helicase starts unzipping, it releases extension, and then the optical trap has to move up far away to keep the force constant. And this motion of the optical trap up away is shown here by this Versal activity, okay, in this force clamp procedure. So I have an animation to show here that gives you a flavor of what we're measuring. So here it's written on the left force versus something, but it's not important. That's time, and this is extension, position of the trap. It's not force. Even if it is written force, it is not. Okay? It's just to give you the idea that what is a position that we measure. This is basically around 100 nanometers. Okay? So we have this noise, and then activity, a burst of activity occurs whenever an helicase binds to the single-strand DNA at that basis of the stem of the fork, and then unzips the residual 100 base pairs. Then it detaches, the hairpin closes back again up to the 100 base pairs position, and then a new enzyme assembles, everything is very fast. Okay? The speed of this enzyme is around 200 base pairs per second. Okay? So the amount of power put into the enzyme by ATP draw is around 2,000 kbts per second. Okay? So let me go back to Grenoble here, 06. Now what you can measure from these traces, here I show you the collection of all these traces, all these bursts of activity I put in a single slide, and they are here, okay? There are 100 trajectories, and every trajectory starts from the position where the enzymatic activity starts, and you see that there is a lot of noise, okay? Now I show you this slide because this is a nice model for molecular transport, for motion, okay, in the presence of what we call steady-state diffusive kinetics. So you can measure the average position at a given time, okay, you can measure the average distance translocated, and you can measure the variance, and according to what you expect from a diffusive process with some advection, okay, what you get is something like this. The average distance covered by the motor is linearly with time. This gives you the rate of 200 base pairs per second. And you can get also diffusivity, okay, in this motion. Okay? So it's a motion is a diffusive motion with advection and diffusion, both things. Okay? So this is very useful, okay? And it gives you useful information about this motor. Otherwise, you have to use other sort of biochemical techniques, but it's a very powerful, the possibility to see this enzyme acting, monitoring the action of the enzyme as a function of time is relevant. And that's the last part of my talk in the last 10 minutes, where I will show you how we can go from work and information to free energy measurements. So there was a remarkable result, something like probably 15, 20 years ago, approximately, which was due to first Chris Jarzynski, then later Gavin Crooks. Other people preceded them, but they were able to put these more in context of making something useful, which is the fact that by measuring these fluctuations, by measuring basically the amount of power that we put into a system, and by measuring the spectrum of fluctuations of that power, you can extract thermodynamic information. So out from irreversible processes, I can extract, for example, the free energy difference between two states. So that's something remarkable. So I show you here the relation by Crookes, which is the following. Suppose I have a system that is thermally in equilibrium at a given value of what we call the control parameter, which would be the position of the trap. And by some reason, I decide to exert mechanical work on my system by changing as a function of time the control parameter in a predetermined way. That's called a protocol, okay? So I change this control parameter as a function of time from initial value to final value. And I start in equilibrium. And here I do not end up in equilibrium. Actually, I may end up out of equilibrium completely. But the important thing is that I start in equilibrium. I do the same for the time mirror, from time mirror image of this protocol. So you put here a line and you do the specular symmetry, and that's what we call the reverse symmetric, the time symmetric process, okay, time reverse process. And you do the same, and now you start from lambda f to go to lambda I, but now here I start in equilibrium lambda f. That's a required condition of this fluctuation relation, or this fluctuation theorem, and I end up whatever it is, out of equilibrium or not. And then what you can prove is that you repeat this experiment infinite number of times to extract the probability distribution of the work exerted upon your system, okay, along this forward process. I measure as well the probability distribution in the reverse process under these conditions, starting in equilibrium at the final value of the protocol, and this independently on the speed of the process. So this is, both protocols are irreversible, induced irreversible processes. The ratio between the probability distributions of the work is exponential of what we call the dissipated work divided by kBt, or if you want, the exponential of the work minus delta G divided by kBt. So this was very interesting because when it was published around 2000, I was at the time in Berkeley, and when we said, okay, there would be a very easy way in single molecule experiments to extract delta G. You just measure experimentally the irreversible work distribution during the forward and for the reverse, and you look where they cross each other. Because when they cross each other, this is equal to 1. So you have two distributions. By hysteresis, you always put more work on the molecule in the system than you get from the molecule to the machine. So there is hysteresis. But when the distributions cross each other, the ratio is equal to 1. And therefore, this gives you automatically W equal delta G. There is a single solution for that. So what we said is, OK, you can extract delta G, which is a true equilibrium free energy difference, which apparently you could only measure by doing a reversible process, by doing irreversible experiments and looking at where distributions cross each other. So we did these experiments that we published in 2005, and this is what we got. I mean, we got these two distributions, and here that's the unfolding distribution of a complex RNA three-way junction structure, which was very difficult to measure the melting, the free energy formation of this structure, because RNAs are particularly singular in the sense that if you want to do calorimetry, which is a traditional way to measure free energy differences, you have to heat up and unfold the structure. But the melting temperature of RNA goes above 100 Celsius. So sometimes you cannot melt them before water starts boiling. So you have an alternative method to look where distributions cross and extract delta G. Okay, and because I have just 5 minutes, I want to explain you how we did for this Maxwell demon, because that closes a little bit the story of our contribution to this project. This theorem has been extended, has been modified, has been applied, and actually these and other papers published back in 2005 are the basis of what we're seeing now, the activity. Many people joined this field because they saw that this was beautiful and useful, okay? So I will switch this part and go to mention this, okay? So that's, I don't know if there's a light of Dimitri. My demon is green, yours is red, so probably that's, so these are black, I don't remember. But this is the idea that he said, I mean, you can, using a small intelligent being that fights against the second law. So it opens and closes a gate in such a way that it separates molecules that have different dispersion of velocities into those that are slow and those that are fast. And this goes against the second law. So is that possible or not? And he already provided the answer, which I will not say anything because it was clear. But the idea is that if you can do this, then you can use such a demon to convert information into work. So here is a classical example of the Szilard's motor. You have a vessel containing one molecule, and then what you do is the following. You see at which side the molecule is, and at that point you close the big vessel in two halves, and then you attach where there is a piston in the middle. So this is a moving wall, and therefore you attach a weight that can be lifted, and you use the expansion, the collisions of the molecule against this moving wall to exert mechanical work against gravity and push up a weight. So you could convert information, which is the observation of which side of your big vessel, your molecule, is, into work. So now we know that the maximum average extracted work by such a device is kVt multiplied by the average information, which for the case of a 2-bit device, like in this case, gives us this Landauer limit kVt log 2. That's the maximum information, that's the maximum work we can extract from such process. There was experimental demonstration shown in the case of single electron boxes by Yuka and Dimitri, and we can do that in a molecule, and that's what we have been doing in the lab. So suppose you take a molecule like a DNA hairpin that can fold and can unfold mechanically. So if you go close to around 15 piconewtons, you will discover that these molecules, these small hairpins, not for the big hairpin, but for a short hairpin of 20 base pairs, you can see that the molecule starts hopping between two conformations, because the barrier that separates the folded and unfolded state is around 6-7 kVts, which by thermal fluctuations in the scale of seconds you can overcome. So you see your molecule hopping between two states, and in our experimental device, in experimental setup, we can operate in two modes. One mode is the so-called passive mode, where we don't move the optical trap, and then the force changes. That's the typical dichotomic signal around approximately 15 piconewtons for such molecule hopping between the folded and unfolded states. So this would be an unfolded state, this is a folded state, and this change in 1 piconewton is the release molecular extension of the 20 base pairs. And you can do that in a constant force let's say in a force clamp operating mode, where you move the optical trap with a loop frequency of around kilohertz to keep the force constant, and then you record the switching distance, trap position. So the experiment that I will show you now is for passive mode, so I don't keep the force constant because it introduces artificial fluctuations or fluctuations that I want to escape somehow, and I will show you what we did. So we have this force hopping signal, okay, in the passive mode, and then we have a daemon that observes which is the state of the molecule. So here in the high level force is in the folded, and here in the low level force is unfolded. So when it's folded, it says, okay, aha. So then what he does is you move the trap close to the other bit, so you release the force automatically, and then you slowly push up the force. And now because the molecule is folded, what it may happen is that the molecule, by thermal fluctuations, unfolds. Okay? But now the first thing that will happen is that it unfolds. So when a molecule unfolds, you are giving it work. When it refolds, it gives you work. Okay? So at the average, because the force is rising up, and because an unfolding process precedes a folding process, you get useful work from the fluctuations in the bath. And the same would happen if the molecule is unfolded. I would push up the force, okay, and then go back the force to allow for it for fold back again and fold back and so on. Okay, so that succession, that time succession of foldings and unfoldings gives you useful work from fluctuation in the bath. We extract heat from the bath, and therefore what we do is we analyze per cycle how many work we can extract out of these transitions, and that's basically Yuka's and Dimitri experiment, and we get this distribution that has, in average, it has some negative contribution, but also positive ones, similar to yours. But of course, we have to check what happened in ours, because our average work is around 0.7 kVt, which is too close. 0.69 kVt is the limit of Landauer. So maybe we were very lucky and we got the Mini-Manger curve where we match it, but we have to reanalyze. This is very recent data obtained by Marco Rivezzi, who is here in front, in the second row in this room. And he did these beautiful experiments. We are still analyzing them. We are doing the experiments at 50 millikelvin, and you got 300. So there is a difference, okay? No, sorry, you got 50 millikelvin, and we're doing at 300 Kelvin. So there is a big difference in temperature, but the basics of the experiment is the same. So I will stop here and thank all people in my lab that made this work possible. And Marco is here, who is a member of the INFERNOS team, and he's sitting here. You may ask him questions as well during coffee break. Thank you very much. I will be pleased to answer you any question. Thank you.
Yes, thank you very much, Felix. Do we have questions? Yes?
Yes, I have a technical question. At which speed you can pull this biological molecule?
So this, with optical tweezers, this depends a little bit on the device you use, but in our instrument we can pull from, let's say, 10 nanometers per second up to 5,000 nanometers per second. Now, that's not the relevant, physically relevant information. I mean, if you want to ask me how this speed correlates to the irreversibility of the process, you must look at the loading rate rather than the pulling speed, how the force increases with time. And this goes between 0.5 and probably 30, 50 piconewtons per second. Depends on the stiffness of the tether you put between the molecules. But that would be the ranges.
And the bandwidth at which you can detect the force is more than 1 kilohertz.
Excuse me?
The bandwidth to detect a force is more than 1 kilohertz, I imagine.
The force you can detect at very high frequency with photodetectors, because our measurement of the force is based on the deflection of the light at the photodetectors. So that would be the responsive frequency of your position-sensitive detectors, which I guess can go to hundreds of kilohertz probably. But you know, we have this force feedback loop and this sort of electronics that averages our data that operates at 4 kilohertz, 1 kilohertz, okay, which is probably enough. But at the end of the day, it's very important to have high-frequency data, but you have to understand that we are measuring the force by looking at the deflection of the light caused by the beat. The beat is in the optical trap. And at the end, everything is limited by the corner frequency of the beat. And the corner frequency of the beat is about 10, 50 kilohertz. It's difficult to go beyond 10, 50 kilohertz. So even if you would have very high resolution in your detection, the beat is not responding. Okay?
And briefly, what is the physics behind this overstretching regime when you have this plateau?
Well, you know, this is one of the subjects you don't want to work, because there are two communities that are against each other. So you have to join one. It's a largely debated problem. Initially, it was thought that this overstretching corresponds to the fact that DNA, which is a double-stranded. So if you take a book of biochemistry and look DNA, and then you ask why it's a double helix, it would tell you no, it's a double helix because, of course, if you have 6 nanometers between approximately 2 base pairs, the thickness of each base pair is approximately 6 angstroms between 2 contiguous base pairs. The thickness of each base pair is 3 angstroms, so you are left 3 angstroms in the middle. So water would fill in. So then, of course, these bases are highly hydrophobic and they don't want to have water inside, so this has to be squeezed. So the only way to squeeze, to pull out water is like with your towel. If you want to pull out water, you have to squeeze it. And that's what DNA does. It's squeezed and the water pops out, okay? So then, if you look at these books, that would be the simplest structure. But it's interesting because you could also think a way of taking out the water with a ladder. So if you had a ladder inclined, then that would be the same. So why it's an helix? Well, helix has higher symmetry than a ladder. Now it seems that when we stretch at 67 piconewtons, we're putting a high asymmetry into the system. So we're breaking the helicoidal symmetry. So it would be natural to think that it goes from the helix to a ladder. So all the bases are tilted along the axis of the force. So this was the initial hypothesis for the overstretching transition. But then it turned out that at the end of the overstretching curve, it was found experimentally that the extension corresponded to that of the single-stranded DNA. So one of the 2 strands, stretched at 67 piconewtons, has the same force. So it seems that if the 2 strands have melted. Okay. So you see here, this extension here. So if I pull, so this is 8 microns, so this is 24,000 base pairs. Now you take 24,000 bases, not base pairs, of a single strand and you pull them, the curve would be like this. It would end at here. So people said, oh no, this is because they have melted. And you have, so this was a big discussion. So nowadays the most accepted model after 20 years of discussion is that it's a mixture of overstretched, so ladder-like, and melted regions.
So if we have no more questions, then we can go to the salle des convivialités and have a coffee break. And we can come back here at 4 o'clock to listen to Yuka Pekola.